All right, this week we're continuing a discussion about CAR T cell therapy, chimeric antigen receptor T cell therapy, and the angle that we're going at now is instead of going through a T cell therapy that's already in clinical trials, I thought it might be more interesting to look at something that's in process that's sort of gearing up towards phase 1 trials in humans and is in the final stages of preclinical preliminary data. And so to lead into it, just a quick recap. Remember, with CAR T, the way that this typically works is you have to have some sort of particular antigen that your T cell is detecting and receptive to. And so when the immune system does this naturally through T cell activation, you typically start off with a helper T cell that recognizes a particular antigen, and that antigen could be from a pathogen like a virus or a bacterium, or it could be from a mutated protein on the surface of a cancer cell. But that antigen is critical because if there's no antigen present that's actually being detected, then the T cell has no specificity. It doesn't really know what to go after. And so our assumption is our helper T cell has found an antigen that it's interested in. It then can communicate that information, right, via cytokine release to activate cytotoxic T cells. Those cytotoxic T cells also recognize that same antigen, but the difference is our cytotoxic T cell is going to release these perforin and granzyme molecules that actually kill the target cell and eliminate it, right? And if we can make so that antigen is something specific to our cancer cell, then makes really good therapy. So this is the natural version of it. This happens all the time to tumor cells inside of a living person. But if someone has cancer that's actually made it to a clinical stage and they're getting treatment for it, that means that something went wrong in this process. The antigen wasn't recognized successfully. And so, we're going to have to take matters into our own hands to some degree. Okay, and remember, to do that, that is the entire point of this chimeric antigen receptor, which is made by taking the cytosolic region of a T cell receptor, right? That's the intracellular portion that will actually turn on a cellular response once it's activated. But the issue is this extracellular portion, that's the part that should be responding to an antigen, but clearly that hasn't happened. Otherwise, the patient's T cells would have already taken care of business and eliminated the tumor. And so what we're doing is generating an antibody, right, that will bind to our antigen. But the catch in all of this is you have to have a target that you're going after, right? So previously when we talked about glioblastoma and lung cancer, it was EGFR, epidermal growth factor receptor. And that was the target, and we kind of just took it for granted that we had a target, we had an antibody that bound the target. We take the antigen binding region from that antibody and fuse it with the sequence that codes for the T cell receptor with some linkers in between to make the whole thing work, right? That sort of glosses over the fact that finding a successful target is probably the most difficult part of this entire process as far as the search goes, right? Doing all of this seems very complicated and it requires some advanced molecular biology for sure, but technically making the CAR, the chimeric antigen receptor, is not incredibly difficult in its own right. Finding the target and getting an antibody that binds to it successfully is a bit more difficult because needle in a haystack kind of problem. There are thousands of proteins expressed by a given cell, and finding one of those proteins that is unique or at least very highly enriched in the cancer cell, that's the difficult part, and that's what we're going after here. All right, so instead of glioblastoma, I chose a target that's being developed to treat melanoma, which is another cancer type that we talked about recently in a slightly different context. And the authors describe the problem really well in this introduction. So the major limitation, meaning the most difficult aspect of developing CAR T for tumors, is identifying surface proteins right, that are highly expressed in tumors but not in the normal tissue. And it has to be a surface protein because those T cells, their receptors can only bind to proteins that are on the surface of the tumor. If we have an interesting protein that is intracellular inside the tumor cell, it's effectively hidden and masked by the cell membrane. T cell won't be able to recognize it anyway. So we have this list of qualities of our target that we're going after. Must be extracellular and expressed on the cell surface. Has to be specific to, or at least very highly enriched in the cancer cell, and it has to be an antigen that has the right qualities that a T cell receptor could reasonably recognize. Okay, so they identify this tyrosinase-related protein 1, right, TYRP1 from here on out. That is the potential target. And using it to treat melanoma, that has been unresponsive to, right, this immune checkpoint blockade. This is actually all connecting because immune checkpoint blockers were where we spent some time several videos back going through those treatments and how cancer cells can evade the immune system. So if melanoma has not been successfully treated by immune checkpoint blockers, we're trying to find a follow-up treatment to attack them via a different mechanism. Right, so problem here. This TYRP1 protein is mostly an intracellular protein within these melanosomes, are organelles within melanocytes, which would be the cells that cause melanoma, right, when they become cancerous. And only a relatively small fraction are trafficked to and expressed on the cell surface. And so the protein is found on the cell surface, but that's not where most of it is. And so they need to develop a CAR T receptor that is incredibly sensitive, meaning very, very high affinity to this protein, because the quantity isn't incredibly high, right? And so high affinity of the receptor is, right, a primary concern, and that's part of the testing that they're going to be going through. Okay. All right, so they develop their therapy. They find a receptor that works, binds TYRP1, and they're testing it in human tumor cells, in vitro and in vivo in a mouse model, and patient-derived tumor models as well that are grown in culture. Okay, so the upside, no major toxicity from the T-cell therapy, and inhibited growth in a significant way for cells that express their target. Okay, so priority one in all of this is find out how many patients have an expression profile that makes this realistic. So this graph is showing patients on the X. If you zoom in close, each one of these tiny little data points is illustrating a single patient. And then this is TYRP1 expression. And so their threshold line in red is effectively saying, they expect that this subset of patients will likely be highly responsive to the treatment because they express high levels of TYRP1. These patients in the middle would probably have some level of response, but expected to be lower. And then these patients, we would expect that this would not be a meaningful therapy for them at all because their tumors just aren't expressing the target. All right. And from there, they tried to see if there was any pattern in effect of separating patients from the immune checkpoint therapy trial. So this data from B was taken from the Checkmate trial, which used, I believe, a PD-1 inhibitor to try to reduce melanoma growth through immune checkpoint inhibition. And so they categorized people by being responders, non-responders, or somewhere in between, right, stable disease. And then they looked at TYRP1 expression, right? And what they found was these two didn't really have a whole lot of a change. For the responders, people that responded to that treatment, they tended to have lower expression afterwards, which suggests that that treatment was preferentially killing off cells that expressed TYRP1. All right, and that turned out to be interesting when you look at this. So number of patients on the Y, TYRP expression on the X, and when you look at responders and non-responders, the data were all over the place. What it really showed was that the previous round of treatment, right, that checkmate trial where they used the checkpoint inhibitors that blocked PD-1, you had a mix of responders and non-responders all over the map of TYRP1 expression. And so there was an association here for responders, virtually no association at all for stable or non-responders. But in the end, the variability was high enough that it looks like, we have patients that might be good targets for TYRP1 treatment all across the spectrum of, whether they were responders or not for previous treatments. Okay. They then further subdivided based on subtypes of melanoma. It's not probably a big deal to go into the details, but these are just 3 different subcategories of melanoma. And as you can see, some of them more so than others. Each bar is one patient, again, TYRP1 expression, and you see a decent number meet the threshold, more so in this uveal melanoma category. But even in the other ones, roughly half the patients or a little bit more met the threshold. And so this is just establishing that within melanoma and even subgroups of melanoma, there are enough patients that have a relatively high level of TYRP1 expression, making the drug even worth pursuing. Because if these were incredibly small numbers of patients, what's the point in even trying to manufacture a drug when there won't really be a good target? Okay. And then last thing I'll mention, melanoma cell lines, they tested a lot of different cell lines that were derived from patients. And same idea. Some had no expression, some had moderate expression, and then a handful showed high enough levels to make them worthwhile as targets. The red arrows here are just indicating the specific cell lines that they use later on in the publication. Okay, so the goal in Figure 2 is trying to identify which specific CAR, chimeric antigen receptor, is capable of detecting the unfortunately low levels of TYRP1 on the cell surface. And so I don't want to spend an enormous amount of time here, but the idea is they developed multiple chimeric antigen receptors, right? So each one of these is just a slightly different variant, right, with different linkers, and they use different binding regions to try to figure out which of these receptors would have the highest efficacy, right? And then, in effect, what they did is they tested, right, each of their different receptors, which is designated by these 3 colors, and they tested them along 6 different cell lines. These are the 2 from the previous image that had relatively moderate levels of TYRP1, and then these 4 are 4 of the cell lines that had relatively high expression level. And on the y-axis, we're looking for tumor inhibition, right? And the goal is identify the antigen receptor that has the highest inhibition at hopefully relatively low quantities. And this x-axis designation, effector to target, basically the ratio of T cells to receptors on the cancer cell. So 5-to-1 favoring more T cells, 1-to-5 favoring more receptors. And as expected, 5-to-1 is going to have the strongest effect because that means that you're giving a relatively higher dose of T cells compared to the amount of receptor. Right? And there was an interesting pattern. Some of these had higher effects on specific cell types, and the trend didn't always hold for others. So even though these 2 both had intermediate levels of TYRP1 expression, the pattern of which receptor was most effective did not necessarily hold true between the 2, meaning there's got to be some other variable in play. For the cells that had high TYRP1 expression, there was quite a bit more consistency, right? Not much variability. When expressed at relatively high levels, almost all the constructs were effective to some degree. All right, the next variable in this is not just the identity of the receptor itself and the binding region, but the linker is a significant part of this, because remember, we're making a little bit of a Frankenstein receptor that's composed of a bunch of different parts assembled. And in order to be activated, we have to make sure that the protein can go through the right shape change or conformational change that it needs to. And so they were testing different lengths and flexibilities of linker. Right? And this long, flexible hinge really just means that they had the greatest success with a linker that had greater length and more flexibility so that the receptor could change shape and bind to the antigen more freely. And so we don't have time to go through all of this for sure, but the gist is they're measuring tumor volume in a mouse model, and each of these are their different chimeric antigen receptor types. PBS and UTD are just two different types of negative control. And as you can see, these are the control values. Tumor volume is going up. And these yellow and orange lines did not really produce anything better than the control, whereas this particular construct, which is the one that had that longer, more flexible linker, slowed tumor growth quite a bit more, right? Delayed it a lot longer. Eventually the tumors did grow, but it certainly had a pretty strong effect during this time range. And the conclusion is it's not just the receptor itself and whether it can bind the antigen. You have to make sure that your antigen binding is capable of really turning on the receptor and stimulating the intracellular signaling aspect of it. Okay, so now they effectively have the construct. They know how to incorporate the linker successfully, and right, we're on our way to having a functional CAR-T. Okay. I also added in this figure because it just shows one more layer of complexity. Again, probably not worth going into enormous detail, but the idea is, we've got our antigen binding region out here. We have our linker, right, connecting it to our cytoplasmic portion. And then all of these different regions are pieces of the intracellular part of the CAR, or the normal TCR, the T-cell receptor. And you can assemble them in different ways. And so what they're basically testing is, this part we know has to stay the same because this CD3 is the aspect of it that we really need to activate. And CD28 is staying the same because that's what inserts into the cell membrane. The variable part that they're testing is whether they put another CD8 connector here or this 4-1BB region. And the details of this, like I said, aren't really worth our time, except to say that there are all sorts of different ways that you can assemble these proteins. They're effectively like a modular sequence, right, where you could imagine linking many, many different components together in different varieties. And since this is very artificial, they're testing which of those varieties are more likely to actually lead to activation of our target. And the main conclusion from this is they found that CD28 as that middle segment in the intracellular portion outperforms 4-1BB. So does that necessarily mean that that's always going to be the case for every version of a chimeric antigen receptor that's always made? No, not necessarily. But you have to test it and find out. And so they're calling this a co-stimulatory signal, meaning if you add that component to the intracellular part of the receptor, you just get more activation once the antigen binds. And we see that down here when that version that contains the extra CD28 performs better at suppressing tumor volume compared to the other constructs. All right, so now that they have their CAR and they can get it inside of T cells, the next part of this is testing it out on a mouse model of melanoma, which is exactly what they did. So we're initiating melanoma in a mouse model at day 0, following that tumor volume over time, cells that are untreated, cells that this UTD is just a negative control, where it's basically CAR T that's just not specifically designed. It's like a negative control CAR T that shouldn't recognize anything on our tumor. Specifically, we don't expect to see an effect, and we really don't. And then compare that with our CD28 containing CAR with a flexible linker that binds to TYRP1. And right, for a while anyway, dramatic reduction in tumor volume, exactly as expected. All right, and then moving on from mouse model, which is interesting, but they want to get as close to human data as they possibly can. And since this isn't actually in clinical trials yet, at least when this paper was published, they are testing this against biopsies that were taken from actual human patients. And so these different cell lines, these are melanoma tumors taken out of actual human patients. And we're testing our same construct, same idea. Tumor grows very well when negative control is applied, and really exceptional suppression for several months using this drug or using the CAR T. Much, much better than in the mouse model, which is fantastic because it's saying, in theory anyway, our cells are even better at taking out human cells than they are at treating the mouse model. And these data seem strong enough that it seems very, very likely that this is probably in or being approved to enter phase 1 clinical trials really quickly. So I assume this is probably a follow-up that we can do in the fairly near future once that data is collected and published. All right. As always, hope it was interesting, and talk to you soon.